So we'll start with the topic inheritance of single gene. Okay, now when we only take a single character, that is the stem height, that is tall, having two contrasting trait, that is tall or dwarf, and we are going to talk about only that single character, we'll say that it is inheritance of single gene. Okay, and these type of crosses which occur when having only one character, okay, or it explains only the inheritance of one character, we call that as monohybrid cross. So as we are diploid organism, anything we need to express in ourselves, okay, pea plant also is also diploid because it is an angiosperm, so it needs to be expressed in two forms. Okay, we'll have to take that in a set. Okay, so what Mendel did, Mendel took a tall plant and he took a dwarf plant. Now what he did, he crossed these two plants. So in the first generation, that is the F1 generation, it is also called the first filial generation. Okay, in the first generation, that is the first filial generation, he got what? He got all the plants which were tall. Now again he did what? He did the selfing of these tall plants. He did the selfing of these plants as to the plants which were obtained in this F1 generation. He crossed those two tall plants and he got the result, which was 3 plants were tall, this is the F2 generation. He got 3 plants which were tall, 3 plants tall and 1 plant dwarf. So he saw that the dwarf character which was there in the parent was not expressed in the first generation. Okay, it was not expressed in the first generation, but again to his surprise, that particular character, that was the dwarfness, did came in the F2 generation. So we can say that this dwarf character was there in the plants of the F1 generation, but it was not able to express itself, and then in the second generation it was able to express itself and it came there. It was present here. Okay, now I will do the cross. This was only a short explanation, and now I will do the cross, how he did the cross, how we do the monohybrid cross. Okay, so we will take a tall plant and we will take a dwarf plant. Okay, we will do it here. Okay, this side. This is wet, so it's not, I'm not able to write here. So what he did, he took a tall plant and he took a dwarf plant. Now the genotype which will be present in this plant will be this, as we are diploid organism, and the genotype which will be present here will be this. Okay, so he took a tall plant and he took a dwarf plant. Now the genotypes will be produced, the gametes will be produced. Okay, the gametes will be produced will be this and this, and here it will be this and this. Now these gametes can combine in any way. This and this can combine, and this and this can combine, this can combine, and this can combine. So in the F1 generation, all the plants in the F1 generation were having this combination, capital T small t, capital T small t, capital T small t, and capital T small t. Okay, and the phenotype, that is the observable character of these plants, were that they all were tall. All plants were tall. Now Mendel was not able to see this mathematical thing, because that was the genotype that was occurring in the genes. He was only able to see the phenotype. He did this mathematical equation, but he was only able to see the morphological character. Okay, so in F1 generation, what he got was all tall plants. Now what he did, he took any of these two plants from here and he did the selfing, that is called the self fertilization. Okay, so these are the gametes which are produced. So these will also produce the gametes. So the gametes here will be the capital T small t, capital T, and here it will be the small t. Here the gametes produced will be capital T and it will be the small t. Okay, now they will unite. This capital T will go with this capital T. So the F1, F2 generation which we got will have this capital T, capital T. The second plant will be the capital T and the small t. The third plant will be capital T and small t, and the fourth one will be small t and small t. Okay, so what he observed in the F2 generation was that 3 plants he got were tall and 1 plant he got was dwarf. Okay, now here I will justify this as to we generally use capital and small letter to express a dominant trait and a recessive trait. Okay, this is the generation, this is the F1 generation, and this is the F2 generation. Okay, and when we have the same see this, how he obtained the tall plant, because he used a true breeding line. So he was sure that the plant would be having this genotype, and he also took the true breeding or the true breed of the dwarf plant in P. So he was sure that it was having this particular trait. Okay, now when we have this capital T and both of these are same, we call it as homozygous. When we have this small t and small t, because they both are same, we also called it as homozygous. But when we have this capital T and small t, they both are different trait. Okay, this is for tallness and this is for dwarfness, so we call it as heterozygous. Now homo means same and hetero means different. Okay, these are some of the basic things you should know, as you have already studied that, but you should know about this. Now when Mendel did this monohybrid cross, he saw that with every trait he tried, that is, we studied various characters like for stem height, pod color, pod shape, he was every time able to obtain this result, that in F1 generation all plants were tall, all plants were having a particular character, and in the second generation the character which was missing in the F1 generation came in the F2 generation. Okay, so he came to a conclusion, or you can say that he told that only one parental trait was expressed in the F1 generation, while at the F2 stage both of the parental trait were expressed. Okay, here we can say that the parental trait, these were the parents. So he concluded that for every character that the parental trait, this is tallness, only one parental trait was expressed in the F1 generation, and in the F2 generation both of the parental trait were expressed. Okay. The contrasting traits did not show any blending either at F1 or F2. If there was a blending between these two, okay, then we would have what? We would have a medium heighted plant. That is the mixing of both the characters, that is tallness and dwarfness. So that is a common sense, we will get medium height plant. But in every generation, in these two generations, we were not able to attain any medium heighted plant. Okay, so it was sure that there was no blending between the two characters. Okay, between the two characters, that is tallness and expression of these two characters. Okay, he proposed that something was being stably passed down, okay, unexchanged from parent to the offspring through the gamete, okay, over successive generation, and he called them as factor. He told that something is going and going in the offspring without changing itself, and he called them as factors. And now we know that these are genes, or we now call them as genes. Okay, gene was so called the unit of inheritance. So we call it as unit of inheritance. Okay, so gene is called the unit of inheritance. Now gene which code for a pair of contrasting trait is known as allele, and as they are the slightly different forms of the same gene. Okay, so tallness and dwarfness. Okay, now this we'll call what? It will call that as allele, because it is having a contrasting trait. Okay, contrasting or same trait, we'll call that as allele. Okay, now from here, from this experiment, he gave the two laws, that is the law of dominance and law of segregation. Okay, now this particular hybridization, that is the monohybrid cross, can also be expressed with the help of a square, and that particular square is called the Punnett square. Okay, that I will do in the next session. So this was all about the monohybrid cross. In monohybrid cross, he took a tall plant and a dwarf plant. Gametes were produced in the tall plant and dwarf plant both. These gametes fused with each other, and there was probability of attaining these plants which were all tall in the F1 generation. In the next step, he took the two plants from the F1 generation and he did the self-fertilization or the selfing of these two plants. And he was shocked, or he was amazed, that the character which was not expressing itself in this first generation, it was able to express in the second generation, that is dwarf. So we will also discuss two terms, that is genotype and phenotype. Now the genetic composition of the cell is the genotype, and phenotype is nothing but the morphological characters of this plant. Okay, so when we talk about this genotype and phenotype, we can see here that in the F1 generation will express itself as, in the F1 generation the genotype was, in the F1 generation, first we will do the phenotype. The phenotype was, all the plants which were observed morphologically were tall. Okay, and in the F1 generation, the genotype was, all plants were tall. Okay, so the genotype was capital T and small t. Now this is what? This is heterozygous. This is not homozygous. So we'll call that all the plants were heterozygous tall. Okay, you can take any letter, any letter in the alphabetical or in the alphabets. You can take any letter from the alphabets, but it is easy when we if we take capital letter for expressing the dominant trait and its small letter for expressing the recessive trait. You can also take different, but that will make you more confused. So it's better when we talk about these hybridization experiments, we take capital letter for the dominant trait and small letter for the recessive trait. You can also take it as D, small d, but that will be confusing when you will describe the result or when you conclude the result. So it's better that when we are taking capital T for tallness, we will take small t for the dwarfness, that is the recessive character. So he saw what? In the F1 generation, the phenotype was all plants were tall, but the genotype was small, heterozygous tall. Okay. Now in the F2 generation, he saw that the genotype was, when we talk about the genotype, it was 3:1. You say this? It was 3? No, this was the phenotype. As to what he observed was 3 plants were tall and 1 plant was dwarf. Now what was the genotype? Genotype, we saw 4 plants. This was this. Now capital T small t, capital T small t, and last was the small t small t. Okay, so this is homozygous tall, homozygous tall. These both are heterozygous tall. And this was homozygous dwarf. So the ratio was 1:2:1. So in F2 generation, the genotypic ratio was 1:2:1 and the phenotypic ratio was 3:1. So from here he gave the law of dominance and law of segregation that we'll discuss in the